Hi, in this video, we're going to talk about compositional analysis. This type of analysis will allow us to compare our clusters in single-cell RNA-seq data between different conditions. For example, if we have some cell populations that we want to track between different genotypes or different disease versus healthy, we're going to use this analysis. Now here, what I want to show you, we already have prepared some data. First of all, I'm going to show you that in the samples, the important part here that we have some metadata added. So you see we have a genotype for each of the 4 of our samples, and we have 2 samples that are wild type and 2 samples that have a mutation here. That is important because these are the conditions we're going to compare our clusters over. Now, essentially what you need to process your single-cell RNA-seq data as discussed in the previous videos. So you need to first process it with cell ranger if it's 10x, perform dimensionality reduction, and most importantly, Leiden clustering, because Leiden clustering generates those clusters that we're going to work with. Now, when you're here already, let me just add the next block, which is called Compositional Analysis. I'm going to add it to the project. And now to the settings. So the first thing is the input, right? This is our input. We have the only one, the cell clustering resolution 0.5. This is the output of the Leiden clustering block. So it is essential that we have a block that outputs clusters. Now, covariates. Covariates are factors that might affect the composition of our cell populations. So here, because we only have one column in our metadata, I'm just going to select this one. But you might have multiple columns in your data that you think will affect the composition of your samples. So, for example, maybe it's a patient ID, maybe it's something else. You want to select everything that you think affects the content of your cell population. And finally, the contrast factor. This is your primary factor. This is the factor that you want to evaluate the difference over. So in this case, it's also genotype. And as you can guess, so contrast factor is always one of the covariates. And finally, the baseline condition is a value inside this genotype factor. It's practically a value in the column of your metadata, which we're going to use as a reference. So in this case, we have a mutation and a wild type, and it's only logical to select wild type as a reference. That's it for the setup. Now I'm just going to click run, and let's wait for the results to appear. Here we go. So what do we see here? These are the values of the differential abundance for each of the clusters that were generated in the Leiden clustering block, mutation over the wild type. Now we have 3 columns. The first one is log 2-fold change. This is just a plain fold change of the number of cells in each population. And you can sort by this column and see what clusters have increased number of cells. But most likely what you want to look at is the relative log 2-fold change. Why? Because sometimes you can see that the numbers change, but they change because there is also a fact. There's also a fact that all cell proportions are linked. So what that means, for example, if 2 of the populations if 2 clusters in your sample shrinked significantly, you might see that all other clusters have now a high number of cells. But essentially, that is not because these other clusters increased in size. It's just the two that we were looking at, they decreased in the number of cells so much that now we kind of see that the all other ones increased, which is not the case. The relative fold change takes care of that, so most likely you want to use that. And if I, for example, sort my data by this column, I can see that actually I have one cluster that is increased, then two clusters that are significantly decreased in their abundance. And then we have a Q value, which is, you can think of it as a P value. So practically it means that like whenever the number is below 0.05, we are more or less confident in the results. Now, how to visualize that? The first way is to use cell group abundance plot, which you can see here. So here we have the clusters right here, then two genotypes. Now, sometimes you might want to play around with it and make it more obvious. What I want to do here, for example, first of all, I think that it's better to have a comparison for each cluster side by side. So for each cluster, I want to have like side by side mutation versus wild type. We can do it in data mapping. We can change this primary and secondary grouping. So I'm gonna say that cell group is a primary group. So now you see on the x-axis I have the cell group or cluster, and now the genotype is going to be the secondary group. So now you see we have our clusters, and for each cluster I have two values, wild type and mutation. Now I also want to tune some other things. So for example, first of all, I want to change the order of the mutation and wild type, because it makes more sense if wild type is on the left. So I'm going to drag that here, and you see now I have everywhere the wild type is on the left, the mutation is on the right. And also, I don't really like these labels because they take two lines, so I'm going to just do that. I'm just going to rotate the labels to the 45 degrees angle. And finally, maybe use a different palette for the plot. So, for example, this one. So this is one way to look at your data. And here we have the abundance in the number of cells and cluster ID for each of the clusters. This is one way to look at it. Now, another way is to look at the composition. So here we have a stacked bar plot with two conditions, wild type and mutation, and each cluster is a segment in this bar. Now, here I also want to change some things. First of all, I also want to change the order. I want the wild type to be on the left and the mutation to be on the right, and I can do it not here, but here. There we go. Now another thing is that I actually want to change the type of this plot. So I want to also see the stream area, which makes it like really nice to see how the clusters change. And last but not least are the colors, because you see the palette that we're using right now, it repeats the colors. So we have like this green for cluster 0, but also for cluster 9 and for 18. So I'm going to go to layers, and here I'm going to select a different palette. I'm going to select the triadic palette, and now you see that we do not have any repetition of the color. So this is now a more easy readable plot that shows us that, for example, this cluster increased in size while these two decreased. And this shows us the relative abundance of the clusters in two different conditions. Now, that is all for this video. Thank you for watching.